Good. So we have a message from the organizing team that we are live, so we can start. So I would like to welcome everybody to this section, which is called Food Processing. My name is Jan Banot. I'm a professor at the Faculty of Tropical Agri-Sciences at Czech University of Life Sciences, Prague. And we all together have this good possibility to join the Tropentag, which is primary or first time organized online, which is quite challenging. Definitely, I can imagine for all organizers. Anyway, very good experience for all of us. So let's start with our session. Our session, we have several, we will listen 5 contributions, and we will start with the first one, which is called Optimization of linear extraction from cassava leaves by ultrasonic and high performance dispersing. It's going to be presented by Sonal Loya from Hohenheim University. Sonia, Sonal, you can start. You have 10 minutes. Please keep the time, and then we will have 5 minutes discussion, and then I will ask the other presenters. So the floor is yours.
Thank you, Dr. Pranath. So I'm not going to repeat the title again, as you've already stated it, and I have a little less time in hand today. So good evening to everyone who's present here. Thank you for being here. And to introduce the topic, I would like to start with mentioning that Manihot esculenta roots, which is cassava root, feed around 800 million people across the globe, majorly in Africa and South America. The other parts of the plant, which is Manipura, cassava wastewater from the detoxification results, cassava leaves, tubers, and root peels are discarded as waste in the rivers and soil. However, these parts consist of high amount of cyanogenic glucosides and micro or macronutrients, and therefore can be used as biopesticide and biofertilizers in agriculture. The cassava leaves have already been proved to have pesticidal activity against 50 different insect strains. This pesticidal activity depends on the cyanogenic potential, which can be related to the linemarine present in the leaves. Therefore, this research focused on the development of a novel procedure for the extraction of linemarine from leaves, which we believe would bring about a technological, bioeconomical, sustainable agriculture, and a environmental benefit. Therefore, the aim of this research was to develop a simple, inexpensive, and less time-consuming extraction method for the extraction of linemarine from cassava leaves. This research was carried out in 3 different steps. The first step was a study of physicochemical and biochemical properties of linemarine and linemarase, which showed us that linemarine was present in the vacuoles of the plant cell wall, whereas linemarase was present in the cell walls of the plant cell. Linemarine has its melting point at 145 degrees Celsius, and it is a solid at room temperature. It was soluble in water, cold alcohol, and hot acetone, and its cyanogenesis occurs at 55 degrees Celsius and pH 6 to 7. Different extraction processes were compared in this study for obtaining a better extraction method and at different extraction solvents. So a solid liquid extraction was performed between a conventional method with sock-slit extraction, and the non-conventional method was ultrasonication and high performance dispersing. The 2 different solvents used were methanol and ethanol. Finally, the optimization of linemarine extraction was performed using box banking design BBD-based response surface methodology. The advantages of this modern optimization technique was that it reduced the consumption of all external factors such as time, chemicals, labor, and capital cost. Also, it resulted in a polynomial equation. The harvest of cassava leaves was conducted between the 5th and 15th leaf lobe of the cassava stalk. These very young leaves were chosen because they have a high amount of linemarine. They're soft and easy to grind. The harvesting was conducted before sunrise to prevent any withering, and they were finally stored in airtight bags at minus 25 degrees Celsius. So oxalate extraction was chosen because it is a combination of the siphon principle and the reflux extraction, and it was used in the pesticidal study of cassava leaves. Ultrasonication was chosen because it was important to inhibit cyanogenesis reaction to obtain linemarine as a whole compound, and studies showed that high intensity ultrasound waves would reduce the enzyme activity, in this case linemarase activity. Finally, high performance dispersing, which I would further refer to as HPD, was chosen because it is also called as a one vessel solution, as all the extraction procedure occurs in just one vessel, which prevents any cross contamination. Also, it involves very negligible labor work, therefore results in the automated procedure. Um, and the and it can be used in single or serial use. Also, the another advantage of HPD was the process that the variation of process parameters was completely possible, such as extraction time, solvent concentration, or rotation per minute. Finally, for obtaining the extraction efficiency, a Pickett paper test was performed, which was performed before and after extraction, through which the cyanide content of the samples were calculated, and finally the extraction efficiency was obtained. The result To begin with the results, initially a standard curve of linemarine was developed between the absorbance at 510 nanometer in the spectrophotometer and the linemarine concentration in micromole, which resulted in an R square value of 0.9996. These are the results from the comparison study conducted between Soxhlet extraction, ultrasonic, and HPD at two different extraction solvents, ethanol and methanol. As can be clearly seen, Soxhlet extraction gave very high extraction efficiency compared to the non-conventional methods. However, for the optimization study, non-conventional methods were chosen because because as if we pay attention to the extraction conditions, the temperature was 35 degrees Celsius in the case of non-conventional method, which was less than the cyanogenesis temperature. The extraction times were really feasible, and the solid and a really low amount of solvent was needed for the process compared to the conventional method, and therefore these were chosen. And when compared between ethanol and methanol, a clear extraction efficiency in the case of methanol was seen, a better extraction efficiency. Therefore, methanol was chosen for the further optimization process. These are the parameters for the ultrasonication process used for the box banking design of experiments. Three parameters, time, solvent concentration, and solid to liquid ratio, were chosen at three different levels, which resulted in an extraction efficiency of 65 to 93%. An analysis of MAPE, which is the mean absolute percentage error, gave the result as 4.7%, which was less than the 10% limit. Therefore, the experimental results could be further analyzed for obtaining an optimized model. To obtain the optimized model, an ANOVA analysis was performed, and the final optimized model was shown here, which is E equal to minus 17.19 plus 5.21 T plus 1.14 S minus 0.06 T into S. Here, T and S are the time and solvent concentration, and E is the extraction efficiency. As can be seen, the third parameter, solid to liquid ratio, had no significant effect on ultrasonication, therefore is not a part of the final optimized model. An interesting observation was observed here, which was the intercorrelation of time and solvent concentration on each other to obtain an extraction efficiency. To elaborate on this intercorrelation, I present to you of RSM curves. These curves show that these curves show the extraction efficiency dependency on time and solvent concentration. At a higher time, at a higher time duration, at lower solvent concentration, high extraction efficiencies were observed, and vice versa. That is, at higher solvent concentration and lower time duration, a higher extraction efficiency was observed. However, if the time and solvent concentration both together reduced or increased, the extraction efficiency was definitely reduced throughout. And this is also represented as a graph, three-dimensional representation here. Finally, the results for the high performance dispersing extraction method. The three parameters chosen here were similar to ultrasonication: time, solvent concentration, and solid to liquid ratio. The extraction efficiency also gave a range of 60 to 95%, and the MAPE value was calculated as 1.7%, which was very less than 10%, indicating that we could further analyze our results. An ANOVA analysis gave the final optimized model, which is stated here, which can, in the final optimized model, we can see that all the three parameters had significant effect on the extraction method, and the extraction method and the polymer also depended on a uh, like the polynomial model shows a two-way interaction and a quadratic term as well. To represent the graphs, to represent the results in a graphitical manner, we as we see uh, when we have a graph with time and solvent concentration, when the solid liquid ratio is constant, it can be seen that the extraction efficiency would reduce if the solvent concentration and the time both increase further or both reduce further, which was an interesting observation. Similarly, an interesting observation was made when between time and solid to liquid ratio, which said that as the solid to liquid ratio and time decreased further and further, the extraction efficiency would improve better. Finally, in the case of solvent concentration and solid to liquid ratio, a parabolic result was obtained where the extraction efficiency would reduce if the solvent concentration and the solid to liquid ratio either increased or reduced, the extraction efficiency would further reduce. However, a really high extraction efficiency will be obtained at a certain limit, which is a lower solid to liquid ratio and a 80 to 90% solvent concentration. Finally, to conclude all the results, biosynthesis of these cyanogenic glucosides, linamarin in this case, would reduce their market cost drastically. It was observed that conventional methods have higher extraction efficiency compared to the non-conventional methods, but then the latter gave better repeatability. They could operate at low temperatures. They used less solvent amount and time duration. And the optimum conditions determined for ultrasonication, where 81.2% of extraction efficiency could be obtained at 20.41 time minutes, 86% of methanol at 35 degrees Celsius and a 40 kilohertz frequency. The similar optimum conditions for HPD, where 88% extraction efficiency was obtained at 19 minutes, 88% of methanol concentration, 0.19 solid to liquid ratio grams per ml, 35 degrees Celsius at 11,000 RPM. These are some of the references used in this study. And thank you.
Thank you very much, Sonal, for your nice presentation and for keeping the time. You exceed just 1 minute. It is great still. So I want to ask if we have any questions to this interesting presentation. We'll look also on chat if we have some messages. I cannot see nothing. Maybe I want to ask you about. Of course, it was not maybe the objective of this research. And what do you think about the costs of the technology? Could be this technology used for standard processing of cassava?
So the aim of the project of the research was to develop a process which could actually be applicable in real time, and so we focus on developing and like on developing the extraction method from ultrasonication and high performance dispersing, which are pretty much available in all analytical laboratories across the world, I guess. And we focus on cassava leaves, which is actually a waste product of the whole of the agriculture. They're either given away as animal fodder, or they are just discarded in rivers and soils, which cause a environmental damage because of the high toxicity they contain. So therefore the expenses of the whole process is uh, very minimal, and the focus was that they could be used in developing countries or in small scale farms, so that so that the because the Lenomarine's cost is really high, so this would reduce the costs drastically.
Mm-hmm, mm-hmm. Okay, thank you very much. If no one from the audience has comments or questions, yes, Ziba, please.
Thank you, Sonal, for your nice presentation. I just have one question. Do you think this extraction method can be also applied for the other parts of the cassava, for example, cassava peels?
Yes, the future scope of the project is that we could expand it to either manipulate the cassava wastewater from detoxification process, or to cassava root peels, or to the tubers as well. However, if we compare the cyanide content in the root peels and leaves, obviously leaves contain a much higher content than root peels, so we would result in better extraction here. But the process can definitely be expanded to root peels.
Thank you.
Good. If there are no any other questions, we maybe can proceed for the other presentations. So again, thank you, Sonal, very much for your presentation.
Thank you.
And we are going to another contribution, another speaker, which is Barbara Getz. Barbara, are you ready? You can unmute yourself.
Yes, I'm ready.
Great, Barbara. If you can please first try to share the presentation with us. Yeah, you can just put it on the full screen.
It's fine?
That's great. Perfect.
Perfect.
So let me to introduce another contribution, which is called, as you can see, the potential of sustainable active packaging solution to reduce food losses in Benin. We are going to listen Barbara Getz, and she is from University of Bonn. So Barbara, you have 10 minutes. And then afterwards, we are going to discuss the topic. Any questions are going to be answered. So Barbara, please, you can start.
Okay, thank you very much for the nice introduction. So today I'm going to present some results of our project, which is called Valvepack. It's financed by the Federal Ministry, as you can see. And we want to present some results of our developed sustainable active packaging solution to reduce food losses in Benin. So as we all know, we have high food losses all around the world. 1/3 of the food is wasted everywhere, every year. And as we can see in sub-Saharan Africa, the food loss is quite small for the consumer, which is different in developed countries. But in non-developed countries, we have more food losses in the chain before at the production to wholesaler and retailer levels. A lot of reasons occur for that. Some are like the main reason is wrong packaging or no packaging and problems during transportation, as you can see on these pictures. So proper packaging solutions can help to reduce food losses and food waste. So this is quite an important issue. And then in addition, we also have the problem about a lot of plastic pollution by a lot of packaging that is based on fossil resources, mainly plastic, as you can see on the pictures, is a big problem, especially in developing countries, because most of the time they don't have waste management systems. So what we aim to do is we want to develop an active packaging solution. So what is an active packaging solution? Normally, food is spoiled by growth of bacteria, of microorganisms like bacteria, fungi and stuff, but also for sure chemical and biochemical reactions are reasons for spoilage. But an active packaging solution is deliberately incorporate components that would release or absorb substances into or from the packaged food or the environment surrounding the food, which can then interact with the food and also with the bacteria that are growing on the food. So it can inhibit and stop the growth or spoilage in pathogenic microorganisms. And we also want to have a sustainable solution. So we need locally available, renewable resources, which are also non-toxic. For example, we are focusing here on phytochemicals, which we isolate from plants that are grown in Benin, that grow in Benin, which are normally used for medical application. So quick to let you know what we did. So we have 16 plant species where we collected leaf materials that we dried, and then we did ethanolic extractions of those with 70% ethanol. And these ethanolic extractions we screened for antimicrobial activity. Therefore, we did the agar-well diffusion assay to have a brief idea if there's activity, and afterwards we did determine the minimal bactericidal concentration, which is the lowest concentration you need to kill the bacteria. We also did an analysis of the total antioxidative capacity and the total phenolic content of the extract, which is also interesting in food spoilage. Then we developed a combination of beeswax, shea butter, and coconut oil, where we included the ethanol extracts. And as you can see here, we prepared 25 square centimeter plates. And I will explain you now what we did with that. So there is a standard so-called the Japanese industrial standard, which tests the antimicrobial activity of different materials. So we have our wax plate, we disinfect, and we add a certain amount of bacterial solution. And then we cover it with a foil. And after a certain incubation, we wash off the bacterial suspension. And afterwards, we determine how many bacteria survived this treatment. Then we determined the number of viable bacteria recovered from this test. And if we have a reduction of more than 2 logs, we have an antimicrobial activity according to this standard. But first, we also had a look at the total antioxidative capacity and total phenolic content, which is quite high. In this presentation, I'm focusing on GA, which is Camelina arborea, and LM, which is Lanea microcarpa, and I will show you what we could do while integrating it into our active coating. So therefore, also I want to show you something about essential oils, because this is also possible for essential oils. Here we see an inhibition zone with Escherichia coli, which is a gram-negative bacteria, and most of them are quite resistant to antimicrobial components. This one's not, against Occamum gratissimum. So we try to incorporate this into our coating. And as you can see in white, we have our inoculum. In gray, we have plastic and our wax mist without active component as a control. And on the right side in pink, we have our wax mist with 10% of the essential oil. And as you can see here, we have a log reduction compared to our matrix of 1 and compared to the plastic of 0.9, which is not enough to show, to tell that it's an antimicrobial active material, but we are on a good way, so we might increase the concentration. The only problem is that the smell is quite strong, so this might not be applicable for a packaging of food. But we also could see for Staphylococcus aureus, a pathogenic gram-positive bacteria, that we could reduce the bacterial growth. Again, we have the inoculum in white, which is the starting concentration. We have plastic and the wax mix as our control in gray. And in pink, we have the wax mixture with 10 milligram per gram and 30 milligram per gram of Lanea microcarpa. And we can see a log reduction, which is, let me show the see, 2.7 and 2.8. So we can say this is an antimicrobial active coating. We also checked for more of the plant extracts we had a look at, and here you can see we have the highest antimicrobial activity with 2.5 and 3.6 compared to our matrix for Gmelina arborea extract. Then, by accident, we saw that if we produce the wax mixture with the active compound and leave it overnight, we have a complete reduction of all the bacteria we inoculated. So if we leave the plates for a bit longer, it looks like the active compound is migrating to the surface and we can kill all the bacteria, which you can see on the right side. Gmelina arborea with 20 milligram per gram within after 24 hours storage before the testing. Yeah, and we repeated that with different storage times. So we stored for 24, for 48, and for 72 hours. And for all of these, we could show this. We have quite high antimicrobial activity of more than 2, which is a very good result. So this mixture is has a big potential to reduce bacterial growth on the surface, at least for Staphylococcus aureus. So as a summary and a conclusion, we can say that antimicrobial active plant extracts can be integrated into a biogenic coating and are still active after a certain time of storage. So, but storage before usage increases the antimicrobial activity of the matrix, which might be due to the migration to the surface to be active. And this sustainable approach can contribute to the resilience of food security in developing countries because they can produce it in their country with their local materials. So the next steps in our project is that we are going to test more bacterial strains. We also want to do combinations of certain extracts. And then we want to produce prototypes and do some storage tests with food products. And then we go into upscaling in Benin and going to make a pilot study. And afterwards, we will also check for the consumer acceptance and see if this project outcome can contribute to reduce the food losses in Benin. That's all from my side. Thank you very much for your attention. I hope this gave a quick view on our project. And if you have any questions, please don't hesitate.
Barbara, thank you very much. Very interesting presentation. Thank you also for keeping the time. Of course, now we have the place to ask one, maybe 2 questions. If you want to ask, don't hesitate, and you can raise your hand also through the application here downstairs in Zoom. There is a small hand in the chat bar, then you can raise it, or you can unmute yourself and you can. I can see on WOVA I have no any written questions. If there are no, I will maybe start with one. I'm interested, or I'm curious, for what kind of food do you think this technology is available or applicable, let's say?
Yeah, so there are different ideas. Because of the mixture, we think we might have a good respiratory water vapor exchange. We are still doing research on that. So this might be applicable without the active compound for other foods, like we are looking at boma, which is the African garden eggplant. The leaves are eaten in Benin a lot. So we think that this might be a good environment. But it also can be used as an active compound where it has contact with food products, maybe for cheese. We don't recommend this for meat and stuff because we might have cross-contaminations. And we're also doing some research on cleanability of this packaging solution so that it can be reused.
Yeah, good, very interesting. Anyway, still one more question. I'm sorry, because you, in your title, you're saying that it's for reducing of food losses in Benin. Why just in Benin? Maybe this technique could be used anywhere, isn't it?
Yeah, that's true. I just said in Benin because we get the products which we are working with in Benin. For sure, a lot of plants are also growing in Western Africa too. But I think I'm not sure if this is going on a big scale on a global, because then you need to farm all these plants. So we are thinking about like a local solution, which is then more for Western Africa, and we are working together with the University of Calavi in Benin. So
Okay.
that's why we are more interested in the regional approach.
Good. Okay. Any other questions? I know that I asked already 2 questions and I said we have time for 2. I'm sorry, but maybe still one question. No? Okay, I don't see. So once again, Barbara, thank you very much for a very interesting presentation. And we are going to listen another presentation. And I would like to ask Sarah if she can unmute herself first and share the presentation with us. Good.
Okay, can I start?
Yes, Sarah. So just I will introduce you. So now we can hear you. It's great. So now I would like to welcome Sarah Fischer. She is from Hohenheim University, as we can clearly see, and she's going to talk about why do agronomists measure leaves and not food? Neglected food security issue. Very interesting title. So Sarah, please, even it's very interesting title, you still have just 10 minutes. As anyone. So please keep your time and you can start.
Okay, thank you for your introduction. So welcome to my presentation. And I'll be talking about why agronomists measure leaves and not food, and why that's a neglected food security issue. So as we know, hidden hunger globally affects about 2 billion people. Most nutrients that are important for humans originate from plant-based foods, whereas plants in turn collect their nutrients from the soil. Particularly interesting in this aspect are elements such as magnesium, phosphorus, sulfur, potassium, calcium, iron, zinc, manganese, and copper, as they're taken up directly by the plant and not produced inside the plant, and they're essential for both plants and humans alike. In food production, there are 2 major disciplines that work side by side to produce and provide enough high quality foods to maintain human health and well-being, and these are, of course, nutrition and agriculture. So food production being very important, agronomists attempt to produce the same amount, yield of foods. And to do this, they often measure the nutrients present in the soil, as well as the nutrient concentration in the leaves, to understand whether the plant has a deficiency, a sufficiency, or a toxicity of certain nutrients, and to correct that nutrient imbalance before it can affect the yield. However, what agronomists often don't measure is the food quality in the food. Nutritionists, on the other hand, who are interested in distributing high quality foods, measure food composition. But doing that, they disregard the plant and the environment and the soil around it, and therefore miss a huge possible cause of variance that is later on seen inside the food composition. Further, there's actually not much indication that the nutrient composition of leaves and of different types of foods are actually associated, since most of the time they're 2 completely different parts of a plant with different functions. Looking at the methods in question and whether they're compatible or not between agronomy and nutrition becomes important when looking at the background of global change. We have an increasing human population with an increasing food demand, coupled with increasing soil degradation, which is already impacting food quality and quantity produced. So if we want to continue producing higher amounts of high quality foods, the methods need to be consolidated and the different disciplines need to work together. Therefore, the one big question remains, are the methods used by nutritionists and agronomists actually compatible, or do they tell 2 completely different stories? For this, we have 3 research questions. Do nutrient concentrations in leaves and edible parts show the same correlations to yield? Do correlations between nutrient concentrations in different plant parts differ between annual and perennial crops, or between storage and generative crop parts? And does soil fertility affect any association between nutrient concentrations of different plant parts to yield? To answer these questions, I chose 2 research areas, Teso South in Kenya, which you see at the bottom of the map, and Kapchorwa, Uganda, which you see on top of the map. Teso South has a low soil fertility and Kapchorwa had a high soil fertility. I worked with 3 different crops, maize, cassava, and matoke, and I collected maize and cassava in Teso South and maize and matoke in Kapchorwa. The reason for this was maize and cassava and maize and matoke were in both areas the crops that were most consumed and the crops that were most cultivated. They also represent 3 different food types. So you have a grain, a tuber, and a fruit. I collected cassava and matoke leaves, the youngest, most developed leaf in the generative phase, and the maize leaf I collected during tasseling. And the food parts I collected whenever they were ready for consumption. I measured the nutrients using a portable X-ray fluorescence spectrometer and compared the different plant parts by field and between regions. And to do this comparison, I used a bivariate linear mixed model with restricted maximum likelihood instead of a normal correlation analysis to include the sampling procedure that we did. So to begin with, I'd like to show you some of the baseline data that we collected. So basically the nutrient concentrations that we measured in the different parts in the different areas. So here we have maize grain macronutrients and maize leaf macronutrients. And macro and micronutrients here are defined by plant nutrition, not by human nutrition. And we can see the 2 different areas where maize was growing. So the high fertility area in green and the low fertility area in blue. And what we can see is that soil fertility has a stronger effect on the maize grain macronutrient concentration than the maize leaf macronutrient concentration. So it impacts the food more than it does the leaf. And the same thing can be seen in the micronutrients, particularly a strong effect for iron between the high and the low fertility soil. And again, we see the same difference between maize grain and maize leaves. Maize grain is more affected by the difference in soil fertility. Here, I'll just show you the levels of nutrients that we measured in cassava and matoke, since I collected them only in one area. So cassava, here you see cassava macronutrients in the tubers, which is blue, and the leaves, which are gray, and the micronutrients. And as we can see, the tubers often have a lower nutrient concentration than the leaves. I'm aware that cassava leaves can sometimes be consumed, but in our research areas, they weren't. So we treated them just as leaves and not as food. Matoke, you can see on this slide here. So again, in blue, the fruits, and in gray, the leaves. And here the nutrient levels between fruits and leaves are more or less similar, except for calcium and manganese. And this strong difference you can also see in cassava. And keep that in mind, it'll come up again a bit later. So first off, I'd like to apologize for the big table. However, here you can see the correlation coefficients between leaf nutrient concentrations and yield and grain nutrient concentrations and yield in the high fertility area and the low fertility area. So every red number is either has a correlation coefficient above 20% or below minus 20%. And what you can see here is that in the high fertility area, the leaf nutrient concentrations, the correlation coefficients of the leaf nutrient concentrations are higher than for the grain, which has very low correlation coefficients, whereas in the low fertility area, it's the opposite. The grain has the highest connections to the yield, whereas the leaves do not. Here is again correlation coefficients of cassava and of matoke. In cassava, we can see that the leaves have a stronger connection, or the leaf nutrient concentration has a stronger connection to the yield, whereas, or correlation, whereas the cassava tubers have a lower correlation to the yield. In matoke, we can see in general just very low correlation coefficients compared to the other crops that we measured. So to put all of that information kind of together, the leaf nutrient concentrations don't actually provide enough information to gauge food quality or food nutrient concentration in the different crops measured. And this is mainly due to nutrient mobility. So in the plant, in the vascular system of the plant, there's the xylem and the phloem. And simply put, the xylem transports nutrients in one direction from the bottom to the top of the plant, water and nutrients, whereas the phloem can move nutrients laterally as well. So for example, if a fruit is developing, the phloem would move nutrients from an older leaf to the new fruit to fill the nutrients. The problem is that not all nutrients have the same mobility inside the phloem. So we have phloem mobile nutrients and phloem immobile nutrients. And some of these nutrients we saw before, calcium and manganese, are very phloem immobile. That's also a reason why, for example, fruits are not always a good source of calcium, because fruits are usually phloem-fed tissue, whereas leaves are more xylem-fed tissue. That's why calcium is more found in leaves than in fruits. What's also important is the function of the edible part, because it defines the nutrient filling time and the pathway that the nutrient takes to get to it. So just an example, if we look at maize grain, we know the filling time of maize grain, it's at the end of maize life cycle, basically, and it's the generative part. If we compare it to the tubers of cassava, it's a whole different story, because the filling of the tuber actually depends on the physiological health of the plant part above it, since it's the storage part and not generative. Finally, we also saw that perennial crops seem to have more of a buffer capacity than annual crops, and therefore show lower correlations, which we saw in Matoke. So the key messages here are that the methods used by agronomists and nutritionists are not compatible. Nutrient transportation and the type of crop part, that is the edible part, are very important to consider. There's differences between the annual and the perennial crops. And efforts should be made to combine nutrient composition databases between agronomy and nutrition, particularly when regarding aspects such as global change. These are my references, and I'd like to thank our donors and partners. Thank you.
Great, Sarah. Thank you very much again for a very interesting presentation. And now we have a time for questions.
I would like to ask a question.
Barbara, please.
Thank you very much for the presentation. Do you think there's going to be a seasonal effect on the nutrients in the leaves? So if you have to looking at different seasons?
As in rain season and dry season?
Yes, exactly, yes.
Well, I mean, cassava obviously is perennial, so grows longer, and so is matoke. Yeah, there probably would be a difference because there's differences in nutrient uptake, whether it's raining or not. I mean, not so much a problem for the annual crops since they basically are only growing when it rains. But yeah, there probably would be a difference. In this case, we didn't observe that because we collected all of the leaves at the same time. So it was all like the same amount of rain they had received. But yeah, for sure, I would guess so, yeah.
Okay. Still, we have time for one question. I will check the VOVA if there is something. So there is, I can see on VOVA one question, if the concentrations per dry matter or fresh matter?
Dry matter.
Dry matter, okay. I have one, last maybe. And my question is quite principal, why the agronomist measure the leaves?
Because when you plant a crop, you're expecting a certain amount of yield. And that's usually also what agronomy tries to do. They try to produce yield. And if you measure the leaf, you can see the health status of the plant. So you can see whether nutrients are missing. That would then affect the yield. And if you measure the leaf, you usually also still have time to correct that. So if you measure that you have a phosphorus deficiency in the crop, you can add phosphorus and save the yield before it decreases. So there's a good reason for it. And it's also like, I'm not advocating to not measure that anymore. I'm more advocating to add, to measure the food as well, to see the effects of the fertilization as well on the food if you're changing anything without realizing it.
So the decision should be made on more complex measurements.
Yeah.
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 Good. So, Sarah, thank you very much for your nice presentation.
Thank you.
We are going to proceed to other interesting topic. So I would like to ask Amina if she can unmute herself and she can share her presentation. Good. So I would like to introduce Amina Ahmed. She is presenting the topic which is called Effect of Cooking Time on Quality Traits of Tanzanian Vegetable Sauces. And Amina is from the University of Göttingen. So Amina, please, you can start and you have 10 minutes as your colleagues. Thank you very much.
Thank you. Good evening to everyone. The topic is a part of the project known as Fruits and Vegetables for All Seasons, FRUVASE, which is led by the University of Göttingen in collaboration with University of Erfurt and other universities in East Africa. The project focuses on improved resource-efficient processing techniques and new market solutions for surplus fruits and vegetables for rural development in sub-Saharan Africa. So here the outline. To start with the background information, the project is specifically dealing with fruits and African leaf vegetables. So how we came up with this tomato and the carrot products in this project. So we conducted field survey to find out what available vegetable processed product in the market or probably in households, specifically looking on the methods of processing, shelf life, sensory qualities from consumer point of view, and therefore we conducted interview with farmers, processors, and consumer, and we found high post-harvest loss and waste of fresh produce, including leaf vegetable, tomato, and carrots, which account up to 70%. And this was from farm, from market, and household itself. And for cowpea and cassava leaves, it was possible to dry. It's a common method they use, but tomatoes and carrot is not common to preserve. So sometimes they feed even animals. So we thought from the consumers and processors and farmers suggestion, they suggested maybe making sauce could be the best option, as in African tradition, women are mostly involved in food preparation, but nowadays they are also engaging in other economic activities. So they're looking forward for convenience, healthy and nutritious food so that they can spare time for other activities. So vegetable sauces are an important part of the diet in Morogoro, Tanzania. They use as side dish, accompaniment with staple foods such as cassava, millet, sorghum, and maize. They make ugali. So they can serve a good source of micronutrients, as the micronutrient deficiency in this region, sub-Saharan Africa, is high. And the nutrients such as ascorbic acid, iron, folic, and provitamin A in the form of beta carotenoids. However, the composition and preparation methods of these sources offered in local markets or household is not always known, seems to vary from one batch to another, even from the same processor. As you can see here in the picture, we have different products from the tomato and carrots, and they just packed in different packaging. So it depends what is convenience for the processor. And therefore, consumer complain about the quality and safety of this produce. Therefore, it is not possible always to assess whether they contribute to nutritious and health diet. Therefore, we adapted the local recipes for making these vegetable sauces from Morogoro. And then we studied the recipes, we studied the preparation method, and we wanted to find out what is missing out or what we can do to improve the recipe and the preparation method in general. So we take into consideration tomato and carrot. So here is a list of ingredients and their ratios. So we had tomato. In the formulation, we also included the baobab. In the local recipe, they normally put starch, mostly from wheat flour or corn, and they also put synthetic citric acid to increase acidity. So in our case, we thought about baobab powder, which was added to increase viscosity and acidity instead of starch and synthetic acid. So also this baobab is a good source of mineral and vitamins, including vitamin C, and it has been tried to replace commercial pectin in gel making. And also we added peanut paste or peanut butter, it depends what is available. So this was added to enhance the appearance or the consistency of the product. From our trials, we found the formulations without peanut, the color and the consistency was different compared to the one with peanut butter, so improve the consistency and the color. And also can help to improve the bioavailability of these provitamins, which are fat soluble. And the rest of the ingredients you can see in the table. And here in the chart for making the produce, so you have your fresh tomatoes or carrots, so you have to do sorting, washing, and draining. We mix with other ingredients, and these can be blended for, in our case, 2 minutes was enough to get a smooth sauce, but depend what is available maybe in the household. The kitchen blender can also be used. So in our case, we use a kitchen blender, which has a sauce mode. The speed is about 7. And then you have your sauce, and the sauce was cooked at 87 degrees plus minus 3 degrees Celsius. So we cooked for 20, 25, and 30 minutes. And this cooking temperature, we started the cooking temperature from the household. So when we measured, we found that 87 was max, plus or minus 3 was maximum, or minimum was 84. And mostly this was cooked to 20, 25, or 30 minutes. So we try to have this temperature range and start with the effect of cooking time on physicochemical property of the sauce. So the sauce can also be packed in glass, as we are concerned about the environmental pollution of these plastics. They cause in the environment the much flooding. So we also had the alternative, what if we introduce these glasses and then we can pack, and then we maybe can be stored and be consuming later. So we, in our ongoing research, we have done the packaging and shelf life studies. So we have the formulation, which up to 6 months, they are still good. So we also forecasting for this type of sauce could be the same, as the pH and titrable acidity is almost similar with the other formulations. So here are the results. So when we see the moisture content of tomato and carrot sauce, you see the tomato sauce had highest moisture content compared to carrot sauce. And this could be due to higher moisture content of tomato itself, which was about 95 and 90 for carrot. And cooking time had a significant effect on the moisture losses. And the higher loss was in carrot. than in tomato sauce. So in tomato, there was about 2 to 4%, and in carrot was 3 to 6%. And this can be explained later. And when it comes to total soluble solids, the TSS we measured in the degree of bricks, the cooking time, the formulation, the formulations had no significant effect on the cooking time, had no significant effect on formulation. However, the cooking time within the formulation, the effect was significant. So when we check the fresh, the total soluble solid, for instance, for tomato sauce was about 6, but the cooking time for 20 and 25, there was an increase in total soluble solids and decrease after the 30 minutes of cooking. The same trend was observed for carrot sauce. So while the total soluble solids increase, because during the cooking, there is a release of soluble solids, including sugar and some acid, which include the water-soluble pectic fraction in tomato sauce. And this, for the case of tomato, could be also the reason why the moisture retention was higher, as water is more bound to this pectic fraction. So maybe the rate of evaporation is reduced. And when we come to titrable acidity, TTA, we found that formulations for tomato sauces, for fresh sauces, was about 0.8 gram per 100 gram, and the cooking time for 20 and 25 also increased the titrable acidity of the formulation. The similar trend was also observed for carrot sauce. So the higher titrable acidity of tomato than carrot could be due to also higher the total titrable acidity for tomato itself, and also cooking release some organic acids or to their degradative and reduction of moisture. So this also could lead to increase of titrable acidity. And generally, the pH of the sauces was within the recommended range for East African standards, 3.3 to 3.7. So it is possible for long shelf life. And when it comes to ascorbic acid, also known as vitamin C, we see that tomato formulations had the highest ascorbic acid content compared to carrot sauce, and the cooking
Amina,
time.
I just have to ask you to go to the conclusion because we exceeded already the 10 minutes, please.
Oh, okay, so the cooking time had effect. And also for total phenols, we see also cooking time had a significant effect. And for the beta carotene, the cooking time had less effect. And there was a retention of these pigments, which reflect the beta carotene, and especially for 25 cooking time. And for antioxidant activity, toward the cooking time reduced it. So in conclusion, we said that total soluble solids, pH, and antioxidant activity of the formulations were similar. Nutrient retention after processing at 25 minutes is promising for the retention of beta carotene and ascorbic acids. And the studied recipes will be further evaluated for shelf life and sensory qualities, and optimal recipes will be identified based on nutrient retention, consumer acceptance, and therefore adaptation of this improved the recipe and preparation method is forecast to reduce the post-harvest loss and waste, to increase food safety and security, improve nutrition, and having the new market potential. Thank you very much. And we also thank the Federal Ministry of Food and Agriculture for funding this project.
Amina, thank you very much for your presentation. So we have very short time for question, if there is any. Do not hesitate to ask.
I have a question.
Yeah.
So I just want to ask you. So it was a really great, interesting work. Yeah. So I wanted to ask you about the timings that you chose, like 20, 25 minutes and 30 minutes. Was it like because like 20 minutes is minimum for the cooking of the sauce, or could you not see for 10 minutes or like more timing?
Okay, so for this time, when we tested that previously, the sensor evaluation, they said maybe if we cook for 10 minutes, the sauce have the uncooked flavor. So it was not as acceptable for the consumer. So we had to increase the cooking time, but also as we want to produce salt that will be kept for long without preservative. So the increase of cooking time is important to ensure that the enzymes are fully activated and the spoilage microbes are also reduced to a level that this sauce will have the long shelf life and then can be kept under room temperature, because for cold storage is somehow difficult in rural areas.
Okay.
Thank you for answering that.
Good. So we are on the right time to switch or to proceed for the others. So Amina, thank you again very much for your presentation. And now we are going to listen the last but not least. So I would like to welcome Ziba. If Ziba, please, if you can try to share your presentation with us. I would like to say that Ziba is going to talk about the impact of processing temperature on drying behavior and quality changes in spinning nettle, which is the Urtica dioica. And Ziba is from Hohenheim University, as we can see now. So Ziba, the 10 minutes for your presentations starting right now. So please go ahead.
Thank you, Jan, for the introduction. Good afternoon, all. I'm Ziba Barati, a PhD student at Hohenheim University. Today, I would like to share a part of our research about the impact of processing temperature on drying behavior and quality change in stinging nettle. I will start this presentation with the background information about stinging nettle as a medicinal plant. I will continue with the experimental plan and showing the laboratory dryer that we use, and afterwards I will present the results and the summary of our research. Ortigodioca, commonly known as stinging nettle or common nettle, is a perennial flowering plant. It's originally native to Europe and it's now found everywhere worldwide. The plant has a long history of use as a source for traditional medicine in all societies. It contains health-promoting ingredients such as flavonoids, phenols, vitamins, for example, vitamin C and vitamin A, minerals such as calcium, iron, and chlorophylls. Nettle are used in different applications, including medicine, cosmetics, food and feed, fiber and textile, and biorefinery. In order to prolong the shell life, the stinging nettle, like most medicinal plants, must be dry after harvest. In addition, drying of medicinal plants is a very important step for easy transportation and easy storage, and also reducing the cost of them. Therefore, the main aim of our study was to determine the influence of the drying temperature on the drying behavior and the quality properties of a stinging nettle. In this study, we developed a basic knowledge about the drying behavior of nettle by recording drying curves. Moreover, the effect of different drying temperature was investigated on the quality of dry products in terms of color, total phenolic content, phenolic acid including caffeoyl acid and chlorogenic acid, carotenoids and chlorophylls. For drying experiment, we use the high precision laboratory dryer, which is designed and built at Agricultural Engineering Institute at Hohenheim University. This dryer consists of two main parts, including micro dryer and bulk dryer. The micro dryer has 3 sample holders, which are equipped with weight balances to measure the mass of the material during the drying process. And the bulk dryer has 2 trays to dry high amount of material for quality analysis. The stinging nettle was dried at different temperature of 30, 50, and 70 degrees Celsius. Air velocity and absolute humidity of the air were kept constant. Absolute humidity was 10 grams per kilogram of air, and air velocity was 0.2 meter per second. And the target final moisture content was 10% weight basis in our study. Here we can see the drying curves of a stinging nettle dry at different temperature. The results indicate that the moisture content decreased slowly until desired moisture content of 10% was achieved. We can see that the total drying time decreased noticeably by an increase of the temperature. The results of color value of a stinging nettle dry at different temperature are presented in this table. We found that the color value of a stinging nettle were substantially influenced during the drying process. In total, the dry sample at 50 degrees Celsius had a better results in terms of L value, B value, and chroma value compared to those dry at 30 and 70 degrees Celsius. The reason can be explained by degradation and oxidation of compounds due to the high temperature of 70 degrees Celsius and the long drying time of 30 degrees Celsius. In this bar chart, we can see the results of total phenolic content of a stinging nettle dry at different temperature. We found that drying at a different temperature had no significant influence on the total phenolic content, and the amount of total phenolic content was ranged from 900 to 1,192 milligram gallic acid per 100 gram sample. These results are in agreement with the reported total phenolic content in other studies. In this bar chart, we can see the results of caffeoyl acid and chlorogenic acid of a stinging nettle dry at different temperature. We found that the caffeoyl acid and chlorogenic acid was significantly influenced by different drying temperature. And dry sample at 70 degrees Celsius significantly had a lower caffeoyl acid and chlorogenic acid compared to those dry at 30 and 50 degrees Celsius. In this slide, we can see the carotenoids of a stinging nettle. The main carotenoid in a stinging nettle is beta-carotene. In our study, we measure beta-carotene and lutein of the dry sample. There was no significant difference in lutein content among dry samples at different temperature, although we found beta-carotene was significantly affected by temperature, and the dry sample at 50 degrees Celsius had a lower amount of beta-carotene, followed by dry sample at 70 and 30 degrees Celsius. Amount of beta-carotene in our study is similar to other studies. In the literature, the range of 25 to 300 milligram per 100 gram were reported. This amount can be varied widely due to the harvesting time, weather, and the age of the plants. We can see the results of chlorophyll in terms of chlorophyll A and B in these slides. And we found that the chlorophyll of a dry stinging nettle at different temperature significantly influenced by different temperature, and chlorophyll and dry sample at 70 degrees Celsius had a lower amount of chlorophyll A and B compared to those dry at 30 and 50 degrees Celsius. To sum up this presentation, I would like to mention the main finding of our research. We found that the total drying time decreased noticeably by an increase in the temperature. We also found that the color value of a stinging nettle were substantially influenced during the drying process. Moreover, drying at different temperature had no significant influence on the total phenolic content and lutein content. In addition, we found that the stinging nettle dry at 70 degrees Celsius contains significantly less caffeomalic acid, chlorogenic acid, chlorophyll A, chlorophyll B compared to those dry at 30 and 50 degrees Celsius. There was a significant difference among the beta-carotene content of the stinging nettle dry at different temperature. From these results, we can see that the choice of drying temperature affects the color and the content of valuable ingredients of a stinging nettle. Therefore, for future studies, the optimum temperature for drying of stinging nettles needs to be determined according to the purpose of the drying process. At the end, I would like to acknowledge German Federal Ministry of Food and Agriculture and FNR Agency for financing this research. Thank you very much for your attention. I'm looking forward for the discussion.
Thank you again very much for your interesting presentation. And we have still some time for discussion and for asking questions. If there are at the moment no questions here, I have one in Wova which we can maybe answer live. The question is if you are going to incorporate blanching to improve the color retention in your study, and if you are planning to use other drying techniques other than mechanized equipment you use.
Sorry again, actually I couldn't hear your question. But
I will repeat it. I'm sorry, maybe I was fast. So the question, first of all, they appreciate your work, and they're asking if you would incorporate blanching to improve the color retention in your study. I mean, if you think about that.
Yeah, actually, it's a good answer. And as we know that the blanching is also a primitive treatment method that also used for drying up especially the leaves for improve the quality. And yeah, for further researches, actually, I'm interested to do it.
And the second part is if you are planning to use other drying techniques than mechanized equipment which you use, maybe in the future.
I mean, because actually the main aim of these studies, I mean, I'm working in the project that the main aim of the project is to use the design and build a new batch dryer for the medicinal plants. Okay. And this study that we did, actually, it's for the gathering the information about the optimum temperature, which is related to the nettle. But if I would like to use another drying technique, yeah, maybe I will use maybe with the vacuum dryer or.
Of course, it depends. If we are going to focus on the quality, exactly, then we have to go to some more advanced techniques than if we are looking to establish maybe technologies which are much more available for the rural areas, then especially in the global south countries, we can look at the solar drying technologies which are based on the renewable sources. They are understand, or they are known as the low-cost techniques. Of course, then everything is the question of the compromise between the costs of the technology and the quality of the final product. Good. Any other questions from the group? No one wants to ask Zipa. Maybe I have one, because I'm quite curious about this beta carotene. It is quite interesting that the lowest concentration or the highest concentration were in 50 degrees. Yeah, this slide exactly. So how you can explain that the lowest concentration was in 50 as compared to 30 and 70 on the both sides?
Yes, sure. As I say that actually the beta carotene are widely varied due to the different reasons. Can be the harvesting time and also age of the leaves and also the weather. I mean, we harvested the nettle leaves in the different times, which is actually close. It was in the 3 days. But I would thinking that this less beta carotene can be also maybe because of the formation of the new compounds during the higher temperature of the 70 degree, or also the longer drying time of the 30 degrees Celsius.
Mm-hmm. So if I understand, based on the statistical test, the statistics shows, statistical significance, significant difference between 50 and 70, yeah?
Yes.
Yeah. Because from the slide, it looks that 50, 70 could be almost similar, but yeah, some differences are obvious.
No,
Good.
50 degree is significantly lower than the 70 degree.
Okay. Thank you very much.
You're welcome.
Any other questions?
Yeah, I would like to ask a question.
Yes, Barbara, please.
Thank you very much. Are you going to test also drying technique on other plants?
Right now, actually, in the project that I'm working, we are drying the nettles and also green oats, and somehow also basic information, drying information also on the peppermint, dragon head, and also trigonella.
And you see differences between those?
Yeah, the composition of the different plants, definitely they are different. And the optimum temperature also can be changed according to the different temperature.
Thank you.
Okay. Any other questions? One more is still possible. Okay, I cannot see any other questions. So we are at the end of our session. So thank you again very much for all of you, all speakers, all those who are visiting us on YouTube or here and participate in WOVA and Zoom. I think we have heard a lot of interesting contributions. So I just would like to invite you, those who are interested, in several minutes, about 40, 42 minutes, we are going to start the poster session at 5 p.m. on the other, let's say, Zoom room. So thank you very much again, and have a nice evening with virtual Tropentag.
Thank
Goodbye.
you.
Thank you.
Thank you.
Yeah, goodbye.
Yeah.
Bye.